So good afternoon, everyone. For a small OGS webinar, it's getting a bit more international. So what we are going to talk about today with Amy is about one project that has been initiated in 2019. The first step of this project has been funded by the Spanish CSIC, the Spanish Council of Research. And we developed this project with Carlota Escutia. ICEPRO is an international collaboration effort for improving paleoclimate research in the Southern Ocean. So it's a project that has started with a couple of partners, but which aims to become bigger. The main question that we had before we started this project was mostly related to the fact that actually most of the existing marine records, because here we are talking mostly about marine, are lacking the last 2,000 years, or when there are some records spanning the last 2,000 years, they were at low resolution only. So you can see on this map a couple of records. This map is not really updated. We need to be updated a bit more. But as you can see that in most of the records, you see it could be in Western Antarctic or East Antarctic. Most of these records actually are qualitative estimates, could be of sea ice, could be of ocean temperatures, could be of primary productivity. But most of them actually, they miss the last 2,000 years. And this is also mostly related to the technical issues when we are coring marine sediment. However, recently, for those who are aware, we recently published in Nature Geoscience by Costa et al. in 2021, the first very high resolution record. So it's in East Antarctica here in the Adélie Basin, where you can see, without giving you any explanation about the interpretation of these paleo records, you can see that we got a very nice and very high resolution record covering the last 2,000 years. And it's about a couple of years of resolution. So actually, except these wonderful records, we still have very few around Antarctica. In addition, we have still many other unresolved issues, which is related to the time resolution, which often exceeds 200 years, except the one that I just presented you, and which leads to some issues when we are comparing a model and data, for instance. Most of the records we have so far are mostly qualitative data, and at best semi-quantitative ones. So we are also lacking quantitative estimates. And we also have a very few edge control for the last centuries and 2,000 years, because when I'm talking about the last 2,000 years, I also include the last decades of the last centuries. And the meaning of the paleoproxy, which is still a very delicate debate, a bit less when we are talking about microfossils, because they have been used for a while, like the diatoms especially, but also foraminifera, radiolarian, silicoflagellate, and dinokist. We have some issues about the meaning of the proxy, the organic proxies that have been developed since, let's say, less than 10 years in Antarctica, like the IPSO 25, which is produced by some diatoms, the GDGTs, which is used also for ocean temperature. And more recently, there are some new developments regarding also the DNA, the sedimentary DNA, and also many other proxies. So we still have many unresolved issues when we are reconstructing paleoclimate around Antarctica. And here I just wanted to quickly show you one of the examples, like the HBI, which is now more and more used to reconstruct the sea ice. And you can see now there are Smith et al. In 2016 has started to calibrate this proxy. So here you can see on this map the diene concentration and the leak with sea ice. And more recently, there was a paper of Elena Forat in 2019, which were focusing on the Antarctic Peninsula. But despite these very recent studies, we still have some big questions like which season mostly reflect the IPSO 25 sea ice proxy? Is it mostly spring? Is it summer? Is it both? We still have some issues about which species are synthesizing these SBI, mostly because we can't find the microfossil remains in the marine sediment and why they are produced. And then when we are looking at this proxy that Amy will develop a bit further in her presentation, how to semi-quantitatively, if not quantitatively, reconstruct a sea ice concentration based on this proxy, and then how to combine with others, and I'm thinking mostly with diatoms. So because of all these questions we have when we were applying our proxies in Antarctic marine sediments, we have been thinking that actually there are a lot of cruises, Antarctic expeditions that are organized every year around Antarctica. And here, this is not an exhaustive list, but it's just to say that there are the Spanish, there are the Americans, there are the Koreans, there are the Italians, the French, Australian, the German, there are also the Japanese. So actually, there are many countries that are organizing their own expeditions, but without connecting to each other. And actually, everyone was developing his own protocol, his own methodology, and his own studies in his own area. However, there are a couple of current working groups who are focusing on Antarctica and especially on the last 2,000 years, like the PAGES Climate 2K, which aims to improve our understanding of large-scale modes of climate variability and the mechanisms and driver of climate change in Antarctica, including the subantarctic and the wider southern hemisphere during the past 2,000 years. There is also another initiative through PAGES, which is the Seaside project, which was the aim of this project is to reconstruct changes in sea ice extent in the Southern Ocean for the last 100,000 years, and to reconstruct how sea ice cover responded to global cooling as the Earth entered a glacial cycle. And then to better understand how sea ice cover may have influenced nutrient cycling, ocean productivity, sea gas exchange, and circulation dynamics. Actually, so these are questions that we are also treating in the PAGES 2K, in the PAGES Climate 2K. And then there is another working group, which is a SCAR working group, previously called UNCLIM 21, which is now UNCLIM now, which aim to answer fundamental science questions related to Antarctic climate variability. And even if it's focusing more on the modern, the near futures and the long-term variations, it's also quite connected to the previous working group. And then I would like to mention also, because you are all here and I think you are all aware about the INSTEAD program, which is to quantify the Antarctic ice sheet contribution to past and future global sea level change. For improving the understanding of climate, ocean, and solid Earth interaction and feedbacks with the ice. And also to better anticipate and assess the risk in order to manage and adapt to sea level rise and evaluate mitigation pathways. So this new working group is also getting bigger and also quite connected to the previous ones. And then I'd like to mention also one European project, which is in its phase 2. The EU PolarNet 2, which aims to provide a platform to co-develop strategies to advance the European research actions. So it's Arctic and Antarctic, but they have a big section dedicated to the Antarctic, and its contribution to the policy-making processes. So actually, when we are looking at all these working groups, they are mostly focusing on gathering a couple of data and the records around Antarctica, either to understand past climate change or also to better anticipate the future climate variation in Antarctica. However, what we still miss is therefore the meaning of the proxy, the calibration, the validation, and how to interpret as much as we can our paleo records for this different period of time. So to advance in our understanding of the Antarctic climate, it is therefore urgent to improve first the coordination of the sampling effort by the different national and international projects and programs, and eventually to develop new ones, and including partnership with private companies. And we'll talk about that a bit more later. Second, to apply common and comparable methodologies to aid the development and interpretation of paleo records, establishing robust calibration based on sample from the water column surface sediment in the last 2,000 years. So which means that we need to go what's going on today through the water column from the locus of production to the sedimentation of microfossils of organic compounds, and also to understand the variability for the last decades, centuries, and the last 2,000 years. And also to improve model simulation. I think it's absolutely needed that we better understand our data sets, our paleo records. We provide a new one, especially for the last 2,000 years, like I mentioned before, and also in different areas that are not converted yet, and to be compared or assimilated in the model simulation. So we developed IcePro, and then actually we raised a lot of questions, which are related, for instance, to based on the different national or international programs, what we can do to improve our understanding of the proxies or the recent climate changes in Antarctica. So for instance, we were wondering which parameters, what kind of equipment we need, which previous, ongoing, and future programs are conducted in the Southern Ocean, and how to interact and coordinate with them. Because of course, we need to sort them out and to make sure that we don't miss anyone. Then which ships are going to Antarctica and can be part of a multi-annual monitoring program, and what kind of international coordination and research proposal structure we can also plan for the future. So initially we had some discussion with a couple of partners from Spain, from South Korea, US, New Zealand, Australia, India, Japan, Germany, and Italy. This project is absolutely not restricted to a small group of people, but aims to be extended and to involve whoever wants to be part of it. We organized the first workshop during the International Conference of Paleoceanography in 2019 in Sydney, where we have been talking about presentation of the past, ongoing, and future planned cruises, to draw a map where we have the different transects, what kind of parameters so we want to look at in priority, how to share a common strategy and methodology of sampling to make sure that what we are analyzing is the same from one cruise to another, which ships are going to Antarctica and can be part of this program, how to establish a first collaboration between the different programs, given that there are some in North America, some in Europe, in South America, in Australia, and so on, or in Asia. Extending the collaboration, because also this is why I think it's quite good that Laura spread this presentation away, because I think we need also to bring a lot of people together. How could we move forward by integrating and compiling the data, and what kind of international coordination and research proposal structure? The thing is that when we started this project, the first workshop was therefore in September 2019, and unfortunately, all the planned cruise for the following winter season has been all cancelled because of the pandemic. So, and given that last year was nearly the same, we had to make a break in this project, and now we are restarting it because there are now some cruises that are planned for the next winter or austral summer season, and this is where we have been also talking with the Laura's group. Nevertheless, we have still started or made the first application of this project by sharing some scientists between different cruises. So the first initiative was in Antarctic Peninsula, where we had a cruise in 2018 and 2020, where Carlota Escutia and Fernando Boyo were both PI of the cruise. And then we have started to collecting samples from the water, from the water column and from the sediment to better understand the proxies. And the same year, so in 2020, we also sent one of our PhD students to a Korean cruise which was led by Kim. And actually, we was doing the same in East Antarctica and western part of the Ross Sea. So this is where we started actually to speak about protocols, about different methodologies, and what kind of parameter we can easily achieve during these two cruises. So for that, we look at several parameters. So this is where it could be interesting also for the biologist, because we were looking at the pigments and the nutrients. We were looking at the diatom and other phytoplankton assemblages. We were looking at the biomarkers. So we were using a very basic equipment based on a filtering system, where you can see here, for instance, one of the filters we used to look at the diatom assemblages. Then we have been coring. We have been checking the different phytoplankton on board, and then using other parameters like CO2, like delta T, data weighting, and so on, which also is quite useful for the physical oceanographers. So now our plan is to continue this project. And so the next step will be, one of them will be in the Antarctic Peninsula, where we are planning again to go there with the Spanish ship, the Drake 2022 cruise. Carlota is among us, so if you have any question related to the Spanish cruise, she will be able to answer. Then we plan to closely interact with some biologists and ecologists using this French Tara Ocean sailing ship, for those who knows. And then we are also developing a connection with a private company, actually Le Ponant, which is a French cruise company. They have been developing a new icebreaker, actually, because we don't have an icebreaker. The Hesperides is not, and the Tara Ocean as well. While the Commandant Charcot is a quite new icebreaker, and also planning to go to the area. So our aim is also to interact with private companies or other stakeholders, and to make a program all together. And we have been also initiating some discussion with the Brazilian colleagues on board the Almirante Maximiano, who is planning also to go to the Antarctic Peninsula. So actually for the Antarctic Peninsula, for the next year, we are planning to coordinate all these cruises together, which is not easy to do, but we have some quite good connections and partnership. But also it's not a project for the Antarctic Peninsula, like I said before, but it's also a project that could be applied anywhere around Antarctica, and you are all welcome to participate. And this is where we initiated also some discussion with Laura for the cruise that she's planning to lead also of the Cook Glacier in East Antarctica on board the Laura Bassi. And we have also been discussing with Julian Müller, who is also going with a German group on board the Polarstern between South Africa and the German base and the Antarctic Peninsula. So just to conclude my presentation, I would like to insist on the fact that IcePro is not either a Spanish project or is not restricted to a couple of people, but which aims to involve everyone who has capacities to go to Antarctica and wants to be part of this calibration, validation work on the different proxies, and also to be connected to the Horizon 2020, to the working groups that I presented earlier, through the pages. Also, we can provide the data because we are collecting a couple of hydrological parameters data that could be shared also with the World Ocean Council. And we are also planning to interact with Future Earth. Obviously, our project could be, in the near future, hopefully, one of the action group of the SCAR, but also interact with the different projects that I presented you, especially INSTANT, where there is this subcommittee, the Paleoproxy subcommittee, which is also connected to the IcePro, even though if in the Paleoproxy subcommittee, it's not only about marine proxies, but it's also about ice core, about the lake, about Mumio, or other archives that we can collect around Antarctica. And then we are also planning to interact with the SCORE, which is the Scientific Committee on Oceanic Research. So thank you very much. And now I let Amy talk about some application that we did thanks to AirSport.
Thank you, Johan. Amy, are you ready?
Yeah, I'm ready. I'll go ahead and see if I can share my screen.
Mm-hmm. Okay.
And this should be now as a full presentation.
Yes, yes.
Okay, great. Yes. So hi, everybody. Thanks for listening today. And I just want to reiterate what Johan said, that we're extremely welcoming the more people who join in and who give us ideas and help us figure out the best way to develop these proxies. The best way is to sample and preserve and analyze and then coordinate the data, the better. So what I'm going to do today is just present two small examples that I hope convince you of the value of IcePro and perhaps entice you into joining us. And I guess the bottom line here from me is that we need a lot more modern biogeographic data if we hope to optimize our interpretation of the paleo records. There are so many cases that we look at the patterns in our paleo data, and we simply can't interpret them with any confidence, certainly not quantitatively, because we know far too little about the modern. And on the flip side, we have many other cases where we're striving to reconstruct a specific environmental variable of interest, but we need to develop the appropriate proxy, and we need to be able to develop that in a quantitative way. And so what I'll do is present just two examples. The first, very focused, that Johan already showed a little bit about, a pre-IcePro project, and the second, more comprehensive, showing you what we did during Powell 2020. And the other thing I want to say is that you can develop these tailored approaches that are very specific, or things that are more comprehensive, and I think both are equally valuable. And certainly the methods that we have so far within IcePro can be adjusted very easily to address something that you might be more interested in. So, please let us know if there's something that we're missing. So I'll step back with a very simple example that focuses on the work with the biomarker-based paleo sea ice reconstruction. And these studies, I think many of you know, I wasn't sure of the audience, but these studies use these highly branched isoprenoids, referred to as HBIs, with different forms. The di-unsaturated isomer, or the diene, is believed to be synthesized by diatoms living within the sea ice during the spring. And then the tri-unsaturated isomer, or triene, is thought to be biosynthesized within open water areas near the sea ice edge. So paleo oceanographers use the ratio of the diene to the triene as a proxy for the ratio of sea ice to open water. And then together, the proxies provide information about the duration of a sea ice season. So the questions are, how do we know this? What are some of the challenges to using HBIs as proxies? And how can an IcePro approach help us refine our use of HBIs? So I'll answer this only briefly by highlighting results from two cruises to the East Antarctic margin, a cruise on the Palmer in 2014, and that was followed up by an Australian cruise on the Investigator in 2017. And in both cases, we were really interested in working along the Sabrina coast, but we employed transect sampling from the ship's uncontaminated seawater system as we transited from Hobart, Tasmania, to our field area of the Sabrina coast. So we were able to maximize our use of the ship during a time period of transit, so about 7 days each way. And in this case, what we did was we coupled HBI work at the stations to taxonomic analysis. So our goal was to try to identify the diatoms that were responsible for the biosynthesis of the HBIs, so connecting the two proxies to one another, the taxonomy and the organic biogeochemistry, and then to connect these data to the strong sea ice gradient that we traversed, which you can see in these satellite images. Johan showed this. This is the results of the initial work published by Lucas Smik et al., and the patterns are really clear. The diene was most concentrated in locations with summer sea ice cover, shown here in the red, and it was present but in very low concentrations in the seasonal sea ice zone and absent in ice-free waters. The triene, which is shown in yellow, was present at all the sites, but it was enhanced in the seasonal sea ice zone as compared to its concentration on either side. and it was linked probably to productivity associated with the retreating ice edge. One of the problems, however, identified by the MVP 1402 work when correlating the diatom counts with the concentration of the diene, or the sea ice proxy, was that the species that we identified as being correlated with the chemistry was a species that I've never seen in the sediments. And so there's this mismatch between the chemistry and what we can see when we look at the diatoms in the sediments. So during the investigator cruise in 2017, Linda Armbrecht followed up on this really well. We added to the data set by building beyond just basic taxonomic data and HBI measurement approach. So Linda was able to work to isolate cells of individual species that were correlated positively with the diene. She was able to culture them and then filter for HBI analyses and also for genomics to identify the genes that were responsible for producing the HBIs. So we can look at sort of the map approach, but we can also go in other directions. So really, depending on what your interests are, we can expand beyond just measuring stuff in surface waters. And then just to follow up on this briefly, I want to illustrate the strength of using the combined biomarker diatom proxies. This is a paper by Varath et al. 2020, which used biomarkers, in this case, something called IPSO-25, the ice proxy for Southern Ocean with 25 carbon atoms, and then diatoms, in this case, just looking at the percent Fregularia curta, which is a sea ice diatom, in 3 cores from the Antarctic Peninsula, Trinity Island, Orleans Trough, and the Eastern Bransfield Basin. And she combined those data and compared it to modern satellite-derived sea ice observational data and then to modeled sea ice concentrations. And here you can see, I know it's a busy figure, but I think it's really a great example of how the approach that we're working to promote with IcePro can advance what we can do with our relatively recent observations or relatively recent paleo records. Now to our more recent work, which is what we conducted during January 2020 aboard the Hesperides as part of the Spanish Antarctic program, thanks to Carlota and Fernando. And what we did was we were able to sample about 150 stations over the course of a month-long cruise transit between Ushuaia, Argentina, and the Antarctic Peninsula. We sampled about every 4 hours for everything that's listed here. And we certainly could do more if there was something specific that someone was interested in. And we're definitely looking for advice on some of the best ways to do these kinds of things. In addition to this kind of sampling, we did conduct visual observations of filtered seawater samples within about 24 hours of collection. So we knew exactly what we were looking at. Not quite real time, but close. And that's because of the time it took for filters that we looked at under the microscope for them to dry. And what I'll do here is just briefly highlight some of our findings. Here you can see the physical oceanographic data that we collected at the uncontaminated seawater intake line. And these kinds of data, they're not new, they're not revolutionary, but they're absolutely critical for the proxy calibrations. In this case, you can see in the upper panel, sea surface temperature and salinity helps us identify oceanographic fronts. For example, the polar front is clearly visible on this SST plot. And we can also see areas of lower sea surface salinity influenced by melting ice, and that's in our most southerly areas adjacent to the retreating ice edge in the Weddell Sea. In addition, we had fluorescence values, which are uncalibrated, but they can be calibrated later by complementary pigment analyses, and these show us the location of phytoplankton blooms, and you can see that off in the easternmost part of our field area. And then later, these can be compared to diatom abundance data, cells per liter. This is in progress now. And one of the things that's most interesting is that the mismatches between the diatom abundance data and the fluorescence point to a pretty big hole in some of our analyses that we'd like to fill. So we're looking for somebody who's interested in this, and that is the identification and quantification of blooms of non-siliceous algae, for example, Phaeocystis, which can be quite important in both the carbon and sulfur cycles. And for these, we really need to improve our ability to evaluate their paleo records. Again, not new, but basic and important background data. These are the nutrient data on top. These are thanks to Manan, who's on the line today. So you can see the silica and nitrate concentrations pictured in the top panel with clear minima showing the nutrient drawdown across the Drake Passage. And this is associated with higher del-C13 and higher C to N ratios produced out of Rob Dunbar's lab at Stanford, a pattern that's been observed and published previously, but to have all of these data together as a unit, I think is incredibly valuable. Adding to these data is this really messy figure, which is the observations of our plankton assemblages. This is based on a qualitative assessment that we made on board and later has been supplemented by quantitative data through light microscopy. And then this has been combined with SEM work by a student in my lab, Jane Karskadin. So over the geographic area that we traversed, we observed 8 distinct plankton assemblages that are clearly delineated across the Drake Passage and into the Weddell Sea by changes in the distribution of all sorts of things: planktonic foraminifera, coccolithophorids, silicaflagellates, cryptophytes, and diatoms. While many of these larger scale patterns are well documented, such as the loss of carbonates as sea surface temperature drops across the Drake Passage, others that are equally distinct jumped out, like the concentration of soft-bodied algae. I'm pretty sure they're cryptophytes just west of the South Orkneys, illustrated in pink. There's a lot to see here, so I'm going to simplify this and just present 2 examples. First, looking at the warmer species up here, and just an example of what some of these things are. Images, the ones that are outlined in green are the ones that are from the SEM, so a big change in scale. So a lot of diversity as we start off, and most of these forms disappear pretty quickly as we move into colder waters. This is a pattern that would be wonderful to trace in the sedimentary record, but several of these groups are really poorly preserved in the sedimentary record. And so the question is, how can IceBuro add to our proxy arsenal by tying some of these ephemeral observations of poorly preserved biota to biochemical signatures? And then how can we look at that in the sedimentary record? The second group I want to point out are these cryptophytes, a bloom just west of the South Orkney Islands. And the question for me was, why are they here? And is this something we can track in the sedimentary record, given their soft-bodied nature? So here you can see just a picture of them. They're just these tiny little green, kind of almost look like green raindrops. And my curiosity about the cryptophytes was initiated by some early work from the Palmer LTER group, this paper by Molina et al., and then coupled to more recent work shown here from the Brazilians, who observed the dominance of cryptophytes associated with a shallow mixed layer driven by warm surface waters in the Gerlache Strait, and that's the figure on the lower left. I've highlighted the area of cryptophytes on our cruise in pink in these relatively warm waters just off the South Orkneys. And so why does this matter? I think it matters because they're so tiny, and because they're so tiny, they are not a favored food group for krill. And as has been recognized for about 20 years, this could have important implications for the polar marine ecosystem as we see cryptophytes potentially replacing diatoms. And since the krill aren't eating them, they are more likely to be fed upon by salps. And this would be a wonderful thing if we could track this in our fossil record, looking at the paleocryptophyte record, to look at changes that may have large-scale implications for polar ecosystems. In addition to all of the analyses that we listed at the beginning, I think there's a lot more that we can do. The more closely that we look, the more questions that we have. And I think that we have a really big opportunity here to expand our toolkit beyond the traditional, which would be, for me, the diatoms that you can see here in the bottom of this figure, Fragillaria abscicylindris, a sea ice diatom that is associated very strongly with the sea ice edge in the Weddell Sea. Other groups of diatoms like this Thalassiocyra here, which I have not seen in the sedimentary record, but it's clearly present in the surface ocean. To other potential proxies, for example, Tetraparma pelagica, this Parmales that Cecilia Morales was able to image from some of our CTD samples. And then this creature on the upper right, I'm actually not even sure what that is, but we have so many other things going on in the surface ocean that I think we have a huge opportunity here to expand what we can do especially in terms of tying these types of observations to their biogeochemical signatures. So the bottom line, I think, is that our ability to address the fundamental question that we're trying to answer, what was the Southern Ocean like in the past, can be greatly improved by a program like IcePro. So thank you for your attention. And on the behalf of Johan and Carlota, I invite you to join us, and we're happy to answer any questions that you might have.